So thanks, Moritz, for organising this symposium, and it's been a wonderful conference, so thank you very much so far. So in adults, neuroscience and fMRI in particular has provided many insights into the processes of visual object recognition. And we know that the ventral stream of the brain down here has this transformation from more perceptual representations in the visual cortex, so things that look more similar, have more similar patterns of activity, through to categorical type representations in the more anterior regions, where it's what things are, what category they're from, that best predicts the patterns of activity that you get. Another tool that's proven really useful in recent years for understanding this ventral visual stream is computational modeling, and in particular deep neural networks designed by engineers to solve engineering problems. It turns out that in many ways they're the best currently available model of some processes in the ventral stream, and they have this sort of hierarchical nature. They're taking things from a sort of a perceptual representation through to a semantic representation as you go through the network. So the question that we wanted to ask is, can we use these tools to help us understand the development of visual object recognition in infants? So to do that, we're going to need to do fMRI in babies, and so we're going to have to do awake fMRI. So we're scanning babies at 2 months and at 9 months, and our general strategy is to just make the babies as comfortable as possible. So they're kind of sitting on this beanbag, they're wearing noise-cancelling headphones. We project onto the inside of the bore visual pictures, so it's kind of like IMAX for babies right above them. And here's an example of one of our subjects watching one of our videos. So we recruited babies both from the maternity ward, a sort of low-risk group, and also from the neonatal intensive care unit. It's a longitudinal study. At 2 months, we recruited 134 babies, and then we followed up 92 of those at 9 months. Now, from previous studies, mainly from Yale and from MIT, we expected a success rate of around 50% in these babies. But it turns out that 2-month-olds, they just really love sitting still or lying still for videos. So in fact, we have a 97% success rate in the 2-month group. That's as in we acquire some data. And indeed, they will stay engaged and stay happy in the scanner for a considerable period of time. So the 2-month-olds, we get on the modal amount of time is 20 minutes of participation, and the 9 months is 15 minutes. You may have noticed, some of you used to MRI may have noticed we're just using the back half of the coil, but we still get good SNR across the brain, and the degree of motion is acceptable. It's a little bit more than adults, but it's acceptable, and we can deal with it. I can tell you all more about that if I had more time. So if we want to separate these perceptual and semantic representations, we're going to need a fairly rich stimulus set to disentangle the representations. And one of the, so we've got these 36 different images that we're presenting. One of the big organizing principles in the adult brain is we know that you get quite distinct patterns of activity for animate and small inanimate and large inanimate objects. There's this kind of tripartite distinction. So we arranged our stimuli so they were equally distributed in those 3 categories. Another thing we know that's represented in these anterior visual regions is category. So we also had 3 examples of each of our different types of stimuli shown in the columns here. All right, so let's see what happens. So let me take you through the way that I'm going to show you the results here. So these are results from the adults. What each point in this matrix is showing you is how similar is the pattern of activity in this anterior category region. How similar is the pattern of activity for 2 different stimuli, the one on the x-axis and the one on the y-axis. I've ordered the stimuli by animate, small inanimate, large inanimate objects here. And so, first of all, if you look down the leading diagonal, what that tells us, you can see there's a kind of red along the leading diagonal here. And what that's telling us is that if you present the same stimulus twice, you kind of get a similar pattern of activity. So that's reassuring. Second thing, you can see this big sort of red blocky structure here. So that's telling us that animate things, in general, have more similar patterns of activity to other animate things, and inanimate things have more similar patterns of activity to inanimate things. One exception for that eagle-eyed of you may notice, rubber ducks, adults saw them like animate things. And the third characteristic you might see is along that leading diagonal, you can see a kind of blocky structure. And that's telling us that three different cats are giving you more similar patterns of activity than a cat and a squirrel. So there's this sort of structure there. Okay, so this is what we see in adults. What do we see in infants? So this is the 9-month-old infants, and you can see an awful lot of that structure is already present. And in fact, if you go down to 2 months, an awful lot of the structure is already there. So we can see that, just the basic sanity check, the leading diagonal is there. So we're looking at, the visually evoked responses here. You've got that blocky diagonal telling you that the category representations are there. And then you've got this big scale thing showing that this representation of animate versus inanimate. Now, here I'm just obviously waving at the squares, but let's actually quantify some of those things. So if we look at this here, so what I've done is I've quantified a set of perceptual features, and these are the sorts of things that we think are encoded in this ventral region from other studies: size, elongation, color, the compactness of the object. And then I've taken some semantic measures, so that's this exemplar, the category, and the tripartite, the things I've already been telling you about. The different colored bars, they show the different age groups. So what you can see is that 2-month-olds are really encoding an awful lot of the same stuff that adults are. Now, you might be wondering if that semantic stuff, obviously things from the same semantic category, they tend to look a bit more similar too. So we can take those semantic measures and partial out the perceptual things, and that's shown in the right-hand plot there. And you can see that you've got this semantic representation right down at 2 months, even when we partial out the perceptual stuff. Now, one of the criticisms, we've done our best with this, but one of the criticisms you might have your own perceptual feature that you think is represented in the ventral stream, and you might want us to study that. So there's a certain arbitrariness when you have to pick particular features. An alternative strategy, which I mentioned in the introduction, is that we can use deep neural networks in the way that deep neural networks represent things as a kind of non-theoretically loaded way to select these things. So just to take you through this graph, we know that the different layers of the neural network, on the left-hand side, the network is more perceptual, the right-hand side, it's more semantic. So both of these graphs, I'm taking you through the different layers of the neural network. For each layer, we're calculating the representation of that layer, and then we're comparing that to the representation of the brain. On the left, I'm showing you the untrained neural networks, and you can see that the untrained network does capture some of the representation in the brain. It captures a bit more of the infant representation than the adult representation, but it significantly captures both. But what's striking is that the fully trained network, this is a network trained with millions and millions of examples. This is a much better model of both the infant and the adult brain. And you can see that the infants also have both semantic stuff towards the right there, as well as the perceptual stuff here. So what this is telling us is that basically 2-month-olds seem to have this representation of visual category. And this really begs the question, where does this come from? Is it learned? So they're 2-month-olds, they've had some visual experience. Or could it be innate? Could there be some kind of evolved mechanism that has baked in some of this categorical type representation? So let's just think about that. Obviously, just want to say one thing. Obviously, we're studying it 2 months, so it's hard for us to definitively answer. But I think there is some strong hints in the data. First of all, if I just take you back to this graph I just showed you. So 2-month-old infants, infant vision is really blurry for the first month, and it's also lacking a lot of its color. They won't be able to see things any distance. By 2 months, they can see stuff up quite sharply up to about 1 meter away, but further than that, they're not going to be able to see it. So the 2-month-olds have only had a fairly limited amount of visual experience. By 9 months, acuity and color are really close to being adult-like, and they've had vastly more visual experience. But you can see there's very little difference between these curves. So it doesn't seem to be going with visual experience. If we also think about how experience would work, I've been showing you the results from this category region. For the category region to be maturing, we'd want a mature visual cortex response as well. So let's look at that. On the bottom here, I'm showing you the results I've already shown you. You can see this category region, it's really mature. But if you look at the top 3 panels here, things are much less developed. They're less well developed in the 2-month-old infants in this visual region. So it actually seems that the higher visual regions are more developed than the earlier ones. And just to quantify that, so on the right here, we're quantifying the category region. On the left, the visual cortex, and you can see that it's less well developed in the infants in the visual cortex than it is in the higher regions. So I think that suggests, that's again consistent with the idea that this category region perhaps has some kind of innate starting point, but the earlier visual cortex is going to work and learn the more detailed visual representations through visual experience. All right, so how could we possibly have this innate category representation? Well, one interesting hint is that in adults, there's some lovely work from Zeynep Segin, Nancy Kamwisher, and others that shows that different category selective regions in adults have different patterns of connectivity to the rest of the brain. And we know that quite a lot of connectivity is set up in babies at birth. But we don't know at the moment whether this pattern of connectivity is something you learn through experience or whether it could be innate and perhaps actually driving these initial categories. So we wanted to look at that. To do that, you need to look at connectivity in neonates. So I don't have time to go into this in detail, obviously, but what we did is we used diffusion tractography to look at the connectivity of different regions that will become category selective. And then we used machine learning basically to see whether you can guess the selectivity of a voxel based on its pattern of connectivity. So we're looking at the ventral surface here. We've got faces in face selective regions in red, houses in green, and a tool region in blue. In adults, you can successfully find those regions based on their pattern of connectivity. If you take the adult classifier and test it on the infants, it turns out you can find the same regions in the infants. So that's shown in the graphs, in the picture, in the renderings there, and also in those bar charts. So this tells us that this long-range category selectivity is already present at birth. And so potentially it could be that that's driving this innate category-specific response. So we're really excited by these results, and I think it's just a new way to probe the development of vision, but more generally, this method of bringing together neuroimaging, computational modeling, I think it just provides a new perspective onto development. It also has potential clinical applications, so at the moment it's really hard for neonatologists to work out which babies are going to develop impairments and which are not. We think that functional MRI, by looking at function, because there's such enormous plasticity in the infant brain, just looking at anatomy is often not such a good guide. So we're excited to develop this as well. So I'd like to give really my heartfelt thanks to the team of people that acquired these data, in particular Cliona Dougherty, who did the analysis of these things, and our clinical collaborators, without which we couldn't do this. And just to leave you with a summary, so I'm suggesting that these categorical representations, we find them at 2 months and we think they may be innate, that the visual details may then be learned later. Hopefully I persuaded you that this method is interesting and exciting, and that also potentially we can use what we learn about infants to guide future developments in artificial intelligence. So thanks very much for listening.
Thank you very much. Very exciting. Any questions?
Hello, yeah, thank you. What do you think is the key morphological feature that is driving this triggering of the animate category? Do you think there is one, or maybe it's multiple features?
So we don't know at the moment, because we've got these 36 stimuli. We can go so far with investigating the characteristics. We do also have a set of videos that we've acquired in the same baby, so maybe we can get a little bit more from that. But it's hard for us to be specific about what features drive our responses. My hunch is that, and the avenue that I'm interested in pursuing, is to think about this really as a sort of a messy statistical thing. That, you've got these rich representations in the visual cortex, but they're probably quite crudely separated. And there'll be a statistical question of, if you were just going to do some crude connectivity between the early visual cortex and these category-specific regions, what connectivity could you wire in that would statistically give you a kind of heuristic as to what was present. And so, really probably it's a messy statistical thing. That would be my hunch, that that's what it is. And that's why I think we need computational modeling to capture what those characteristics are. And then we'd like to further look at, measure what they are with future experiments.
Yeah, so just one last thing. What about the duck, the plastic duck? What was the results from that duck comparison?
The duck is seen by infants and adults as an animate thing.
Okay, so that was also, okay.
With hindsight, I don't know whether that was a clever stimulus choice or a really stupid stimulus choice. I alternate.
All right, thank you.
Hi, so this is cool stuff. I was thinking about, so when you were looking at comparing it to the neural networks, one of the things that sort of jumped out from the graphs is that the best fits for the low-level visual features are from these sort of middle convolutions in the neural network. And it got me thinking about this, about the work that's been going around in vision science lately about mid-level features and text forms and things like this. And I was wondering if you think that that's what's driving this. That is basically what the animate-inanimate distinction is based on is not necessarily whole objects, but something like mid-level visual features like text forms.
I think that's a great suggestion. I mean, so we're seeing an animate-inanimate distinction. Maybe it's relevant to the previous question as well. We're also seeing a categorical distinction in the infants. So the infants can tell the difference between a cat and a squirrel. Their brain representations can tell the difference between a cat and a squirrel. So it's a bit more subtle than a sort of really crude feature that you might just use, or cruder features. It has to be a bit more subtle. I think that I'm going to kind of give the same answer again, I guess, that, this sort of messy statistical type stuff is probably where it's at. So that, and I think it's really interesting. I doubt that what we're seeing is really a categorical representation. I doubt they have a categorical representation of squirrels and a categorical representation of cats, but what they have is some kind of manifold that allows them to separate different classes of what, that on that manifold, it kind of separates different classes while pulling together different examples of the same thing. So it's a kind of initial starting point that, helps them separate, that helps them find these invariances. So those sorts of messy statistical things would be what would be useful for that. But we don't know at the moment. It could be a categorical response. Who knows? Maybe they've baked in different animal types.
Just to follow up on that, I don't remember exactly, but the animates that you used, you had the cats, the squirrels, were they all mammals, basically?
Seagulls and crabs.
Okay, and do you think that there's category by, like, essentially which part of the taxonomy you're looking at?
I don't know. I mean, what...
I'm just curious because I think...
do adults have that? I don't know.
Because I was thinking about the rubber ducks because they're a weird case, right? The shape of them is nominally the shape of something bird-like, but the texture is completely wrong. And the fact that they're categorized with other animates, I was curious, are they categorized with all the other animates, or are they just categorized with the things that have more similar shapes or the things that have more similar textures?
It's hard to tell. I just don't know how much we should be reading into the fine detail. And it's not that we haven't done it, so I don't know the answer. I mean, just going back to there's some, on the features, what kinds of features? There's some lovely work from Martin Hebert, finding like the 60 different dimensions that adults seem to represent in their ventral visual stream. And they're really, they're a weird mix of things. Some of them are kind of more perceptually labeled things, and others are very class, food stuffs versus whatever. So I guess that would be a very interesting place to start as a basis set in future studies to try and tease apart. But that kind of thing, or the text form type, those mid-level visual features.
Maybe we have time for one more question. Yeah.
Hello. Mine is a clarificatory question on the method. So I wish to understand how did you exactly combine the fMRI data to generate the model in those graphs that you showed of the untrained and the trained. So what data points did you use and how did you generate the model, basically?
Apologies, sorry for skipping over that. So, you get this in the adults, in the humans. We get these representational dissimilarity matrix by each point is just actually a correlation of a pattern of activity due to one. The pattern of activity you get due to one stimulus and the pattern of activity you get to another stimulus. You can do the same sort of calculation to get an RDM for the activation in a neural network. So if you've got an activation profile to a cat, an activation profile to a squirrel in a layer of a neural network, so just the values, you can correlate those two things together and say how similar is the cat and the squirrel's neural activation in this layer, artificial neural network activation in this layer. So you can get an RDM for a given layer of the neural network. And you can then do a second level correlation where you say how similar is the neural network's representational dissimilarity matrix to the brain's representational dissimilarity matrix. And that's how each point in there is that second layer correlation. So the RDM space is a kind of common currency where you can compare things, which obviously isn't possible when you've got there's no correspondence between one neuron in an artificial neural network and one voxel in a brain. There's no sort of, yeah. Is that clear?
All right. Thank you once more, Rhodri. And